your record.
Okie dokie. I will share the recording with the office of Dr. Iwasaki prior, but I think we have a go already to record, yes.
A go, okay. Okay, thank you so much for all of that. So, well, then I will very briefly thank you again for being with us and for being with the Mexican Society of Immunology. We are by far much smaller than the AAI, of course, but we're still a bunch of people that really like immunology and do very good work in immunology. And I hope you will be able to share more of the AI activities and more of you with the Mexican Society of Immunology, because we really have a lot of very young people and very talented young investigators working with us. And with no further more than this, I would like to leave Marion to introduce you. And so Marion, go ahead and
yes,
welcome our guest.
Thank you so much. I would like to thank Mario, who is a PhD student in your lab, and he made that introduction. So that was really nice. Mario is a Mexican immunologist. So without further ado, I will introduce our speaker of the day, Dr. Akiko Iwasaki. She is a Sterling Professor of Immunobiology and the Director of the Center for Infection and Immunity at the Yale University School of Medicine. She's also a Howard Hughes Medical Institute investigator and is at the forefront of several long COVID investigations. Professor Iwasaki's research focuses on the mechanism of immune defense against viruses at mucosal surfaces, which are a major site of entry for infectious agents. Her team currently focuses on studying long COVID through involvement in multiple collaborations, including a cross-sectional cohort with Dr. David Putrino in Mount Sinai School of Medicine, a longitudinal cohort that's called Yale Listen, and the Yale Recovery Study on the impact of vaccination on post-acute sequelae of COVID with Dr. Harlan Krumholtz. Thank you very much, Dr. Iwasaki, for being here, and the floor is yours.
Thank you very much, Marion and Yvonne, for the opportunity. I'm very happy to be here. Okay, I'm going to share my screen. Okay, you can see my screen, yes? Yeah. Okay, great. Thank you. So today I'd like to focus on our work on the immunology of post-acute infection syndromes, which includes long COVID. And it's really come to the forefront of our, society, where people who get COVID, not everyone recovers. And long COVID has really shined light on this, but long COVID is not unique to COVID in that there are many causes of chronic diseases that end up causing many different organ system involvement. And we started out with this simple question of how do viruses cause disease? And over the years, we've been looking at, for example, humans that are exposed to multiple different viruses all the time. And there are also endogenous retroviruses that occupy 8% of our genome that don't cause any infectious particles, but can express proteins and RNA and cDNA. And there are also viruses that become latent within us. There are many herpes viruses that become latent. They don't cause any diseases, in healthy individual, but when there's immunosuppression, you get reactivation of these viruses. And there are also acute viral infections. And that definition of acute viral infection, for instance, that's applied to SARS-CoV-2, may not be accurate as we're learning more and more about the persistence of these RNA viruses that are supposed to be cleared within a couple of weeks. So one way in which viruses can cause disease is through damage that is mediated by the life cycle of the virus. For instance, if it's a lytic infection that lyses the cells that they're infecting, that alone can cause the damage to the tissue and cause disease. But often what you see is the immune responses that are also leading to diseases within an individual. And, often viral infectious diseases are a combination of these two factors, sometimes more on the virus cause and sometimes more on the immune cause. And it's very important to study these underlying causes of disease if we want to intervene with these infectious diseases. So COVID has really sort of highlighted these post-acute phase of infectious diseases that lead to so-called medically unexplained chronic diseases, such as myalgic encephalomyelitis and chronic fatigue syndrome. And what is underlying and what are the mechanisms that are leading to these chronic phases still remain unclear. And that's something that we're trying to understand, is what are the root causes of these diseases. So, for instance, SARS-CoV-2 is the most recent pathogen to join this list of many pathogens that cause medically unexplained chronic diseases, such as those caused by Ebola virus, dengue, polio, SARS, chikungunya, and many others. There's also non-viral pathogens such as Coxiella, Borrelia, and Giardia, which are bacterial and parasitic pathogens that are also known to lead to this chronic sequela. And I had the opportunity and the pleasure of working with Jan Chotka, who himself has ME/CFS and has done enormous amount of research. And that's something that I've been really appreciating throughout the pandemic, is that patients who are suffering from these diseases are really the experts of these diseases, and they have so much background knowledge as well as lived experience that we can learn from. And so how are these post-acute infection syndromes manifesting in people? There are over 200 symptoms that are reported for long COVID, but there are some core symptoms that many people share. And these include severe fatigue, as well as exertional intolerance, and many neurocognitive dysfunctions, as well as flu-like symptoms, sleep disturbances. This is very common also in these post-acute infection syndromes, dysautonomia, myalgia, and arthralgia. The reality of these long post-acute infection syndrome really is pretty stark when you look at the global prevalence of these diseases. It's estimated that at least 65 million are suffering from long COVID. And this number is likely an underestimation because we're not really counting all the infections anymore. There's also an estimate of about the same amount, 65 million, living with ME/CFS. And this was a pre-pandemic number. But it's likely much more now that we have long COVID patients that are also qualifying for ME/CFS. And the reality is that more than a quarter of the ME/CFS patients are bedbound or housebound. And these diseases can strike people of all ages and background. And these types of diseases occur more frequently in women than men. And not only is this a health crisis, I think this is also becoming a social, economic, and national security problem, as so many sector of the society is being affected by these diseases. So we are considering that there are at least 4 possible root causes of these PAIS, what we call post-acute infection syndromes. One is the reservoir of virus or bacteria or parasites that cause these diseases, that in some people, somehow these viruses and pathogens tend to be remaining and likely replicating to some extent, because after many years of initial infection, people have evidence for viral RNA and protein throughout the body. There's also the possibility of autoimmunity that's caused by an acute, triggered by an acute infection. And this is known to happen for things like multiple sclerosis after Epstein-Barr virus infection. But SARS-CoV-2 may be triggering certain types of autoimmunity, and there is an increase in the incidence of autoimmunity after COVID. There's also tissue damage and dysfunction that can happen. And this is likely mediated by chronic changes or epigenetic changes in long-lived cell types such as macrophages and microglia. And that can impact the host in a prolonged manner, even though the inciting agent is already cleared. And finally, we see evidence for latent virus reactivation. These are, many of these are herpes viral family members that reactivate in people with PICE. So I'd like to share some evidence for all these 4 root causes. And a first study which hinted at these causes are coming from our collaboration with Dr. David Petrino from Mount Sinai School of Medicine. So Dr. Petrino has a big clinic that sees long COVID patients, probably in the thousands. And he's been at the forefront of caring for these patients throughout the pandemic. So we're very fortunate to collaborate with him and his team, Jamie, Laura, and Dana. And from the Yale side, we have 6 trainees that also contributed equally to this study: John, Rahul, Peiwen, Jill, Jeff, and Sasha. And Aaron Ring and David Van Dyke contributed their the platforms. Aaron contributing the rapid extracellular antigen profiling, which enabled us to look at antibody reactivity to autoantigens and viral antigens. And David, with his machine learning techniques, helped us figure out which immune factors are important. So this Mount Sinai Yale or My Long COVID study recruited participants who have long COVID, as well as controls. So convalescent controls, these are the people who had COVID at the same time, around the same time as the long COVID patients, but they recovered fully. There were also healthy control participants who never got COVID. And then we had healthcare workers from Yale that also contributed, as well as an external long COVID participants. These are the Yale pulmonary long COVID clinic participants who served as important validation cohort. And from these participants, we did an electronic medical record and symptom survey, as well as flow cytometry, and then this REAP that Aaron developed, which is a human exoproteome library screen. We did some SARS-CoV-2 antibody profiling. We did a serum immune analysis, which allows us to map the peptide specific for epitopes that our antibodies are detecting, and then the plasma proteomics. So the cohort that we studied, the long COVID and the convalescent controls, we tried to match them as much as possible. Their age group is about 45, 38 years of age. Sex is female dominant, and this is true for many long COVID clinics. There are more female patients than male patients. We also focused on acute COVID severity being mild, because 90% of long COVID patients suffer from after mild acute infection. And then we also looked at days from acute COVID-19. So long COVID and convalescent controls, they are around 400 plus or minus days post-acute infection. I'm going to highlight the features that were unique in people with long COVID. So the orange is always the healthy control. The convalescent control is in this sort of light orange color. And then the purple represents the long COVID participants. So we saw increases in non-conventional monocytes, as well as the activated version of these monocytes, and also decreases in circulating conventional dendritic cell type 1. And increases in activated B cells and double negative B cells. Double negative B cells have been implicated in different autoimmune diseases. We also saw that the T cell compartments were affected, such as reduced circulating levels of central memory CD4 T cells, and increased percentage of the exhausted T cell population, both CD4s and CD8s. And we also, upon in vitro stimulation of these T cells, we saw that there's elevated levels of cytokine production, such as IL-2, IL-4, and IL-6. And when you look at the T cell compartment that secrete both IL-4 and IL-6, they're pretty much confined to the long COVID patients. So this is interesting because this sort of a weird type 2 immune cells, CD4 T cell feature that we are detecting in people with long COVID. We also detected viral antigen-specific antibodies. And what we saw was that there was an increase in the levels of anti-spike antibody, IgG, in patients with long COVID. And we only included people who had 2 doses of vaccines just to ensure that exposure number is similar. And this was the case for the full-length spike and S1 and RBD reactive antibodies. So we suspect that there may be a viral reservoir that's driving the elevated levels of spike-specific antibody. And so far, there are already 2 other groups reporting similar elevation in anti-spike antibody in people with long COVID. So this is kind of a consensus that we're seeing in the field. In terms of the plasma levels of hormones and cytokines, we see that there are some elevation in complement as well as chemokines and cytokines in people with long COVID. But a most outstanding difference was seen in the level of cortisol. So cortisol levels were reduced in people with long COVID. And when you think about the cortisol, obviously it's a diurnal hormone. So we wanted to ensure that the time of collection of the blood was around the same time, in which case it was. And then this is the level Z-score for the cortisol. In patients with long COVID, they have much reduced levels. And because this was so striking, we wanted to validate this in an external cohort, which we were able to measure from the Yale Pulmonary Clinic. And they also had reduced levels of circulating cortisol. So this led us to examine what may be the upstream defects that may be leading to reduced cortisol level. And as you know, the cortisol is a tightly regulated hormone that's secreted by the adrenal glands in response to the ACTH hormone that's secreted from the pituitary, which in turn is responding to the hypothalamic CRH levels. So the pulsatile stimulation from the hypothalamus to the pituitary, acting on the adrenal gland, is regulating the level of cortisol. And when the cortisol levels are produced and high, it has negative feedback loop, which suppresses further production of cortisol from adrenal glands and the ACTH and the CRH. So it's a very nice negative feedback loop through the cortisol. So because we see lower levels of cortisol from long COVID participants, we wanted to understand whether there is a compensatory increase in ACTH to counterbalance this low level. And that's not what we found. In that people with long COVID had similar levels of ACTH as those with control and healthy control groups. So this suggests that there may be a dysregulation at the level of the pituitary or the hypothalamus. And that's something we're looking into using MRI and other measurements in these participants. But what about these other ideas? Latent virus reactivation, for instance. We looked at this using the REAP strategy that Aaron developed. So he included 200 plus viral antigens in the REAP library. And so we were able to detect that there are 3 antibodies that IgG reactivities that were elevated in those with long COVID. And this is EBV specific antigens, P23 and GP42. as well as varicella zoster virus, GE reactive antibodies were elevated. And this is not because of some kind of mononucleosis or acute infection, because using the Serimune, we were able to confirm that there's no mononucleosis happening in these patients, but rather, and then also the latent antigen reactive antibodies were very similar between the groups. So this suggested that this elevated antibody, IgG, against these lytic antigens may indicate recent reactivation of these viruses. And because this was very important to validate, we used two other orthogonal approaches. One is ELISA, one is Serimune, and found very similar responses, not depending on which strategy we used. For instance, the P23 specific REAP antibody score, you can see that long COVID participants had much higher levels. And that is also the case if you look at the GP42 reactive antibody against this particular linear epitope using the Serimune strategy. And then this is looking at GE reactive antibody in patients. And what's also interesting is, as I mentioned, that there are these IL-4, IL-6 double positive CD4 T cells that are uniquely elevated in patients with long COVID. And that level seemed to correlate with the antibody reactivity to EBV lytic antigens. So this suggests that there may be a link between these type 2 immune cells and the reactivation of the virus. Whether one is driving the other, we don't know. And whether there's any direct correlation, we don't know yet. But there is this interesting correlation between the EBV reactivation and these cytokine secreting T cells. And so then David Van Dyke's trainee, Rahul, used some machine learning algorithm to come up with a handful of immune factors that can distinguish people with and without long COVID. And these are the factors that are indicated here. Cortisol, lower levels of cortisol, CD4 circulating central memory cells, as well as CDC1, this reduction in these factors, as well as galectin-1 and EBV reactive antibodies. So this told us that long COVID, it can be explained by these handful of immune factors, or at least distinguished, and that these may be some of the key drivers of disease. But to say that one is causing the other, it requires a lot more research. So then we went on to do a sex disaggregated analysis of the same data set. And this was important because, as I mentioned, this disease is more predominant in females than males, but we haven't really looked into immunological differences. So this is what this analysis is really demonstrating. And this work is done by this group, Julio Silva, Takehiro Takahashi, Jamie Laying, and David. And it's currently in medRxiv. To do this analysis, we separately looked at the symptom burden and organ system involvement in female versus male participants with long COVID. And you can see that both symptom burden and organ system involvement is higher in females than males with long COVID. And Julio also looked at distinct symptoms that affect male versus female patients. So this graph is very informative because if you look at the top of the graph, the symptoms that are being reported by males and females are similar, such as sleep disturbances, disorientation, indigestion, reflux, and so on, fatigue, nausea. These are very similar between males and females. However, when you go down this list, down here, there are female predominant symptoms, that there are many more females that reported these symptoms than males. This includes things like dizziness, body temperature issues, cough, throat, and chest pains, and headaches, and so on. The most male predominant symptom turns out to be sexual dysfunction. And the female predominant symptom is the hair loss. So just by looking at these symptoms alone, you can already tell that there are different organ involvement and potentially different drivers of these diseases. In terms of the immune cell differences, we see that females with long COVID had elevated levels of the CD4 exhausted T cells, interferon gamma secreting T cells, as well as these IL-4, IL-6 double positive T cells. And they also had mainly this EBV reactivation happening. It's mainly happening in females rather than in males with long COVID. Long COVID males also have elevated NK cells, TGF-beta, and APRIL. These are very distinct factors from what I just showed you for females. So then Julio looked at circulating hormones and realized that females with long COVID have lower levels of testosterone compared to the control females. And then males with, of course, males in general have a lot higher levels of testosterone as expected. But he also honed in on the role of these hormones in predicting long COVID status. And he found that this per unit change in testosterone was the top predictor of long COVID status in females. And cortisol reduction is also one of the features that can distinguish long COVID participants, as I already indicated in the sex integrated study. But if you separate the females and males, then you really see the sex hormone testosterone being the top predictor for long COVID. And conversely, males with long COVID had reduced levels of circulating estradiol, and per unit change in the estradiol turned out to be one of the top predictors of long COVID status in males. And cortisol obviously also does that, but it's not as robust a predictor as the estradiol. So this indicated that there's definitely some significant endocrine dysregulation happening in people with long COVID. And what about autoimmunity? Well, I haven't said much about autoimmunity because when we had this sex integrated analysis, we didn't see a significant change or differences in autoantibody levels. However, so Kayla Sa, she's a postdoc in the lab, did this experiment where she purified IgG from people who are healthy, convalescent controls, acute severe COVID. This is hospitalized patients with acute COVID and long COVID. And she passively transferred these into C57-6 mice, just wild type animals. And then she did a battery of behavioral tests. She did not find significant changes in many behavior tests, except for three that are unique. And so this is, one of them is the balance and coordination activity. So when she looked at the rotarod assay, and she put the animals that are injected with antibody, either from healthy controls or convalescent controls or acute COVID or long COVID, onto these rotating rod, and she measured how long it takes for these animals to fall. And so the quicker they fall, the less they are able to balance and coordinate their movements. And that's what you see here with the people with long COVID have IgG from people with long COVID. It makes these animals unable to hang on to the rod, as well as those who are from healthy control. Also, she measured grip strength and then found that patients with long COVID patients' IgG, when injected into animals, these animals had much faster time to releasing them from the grid because of their weakened muscle strength. And finally, she found that the hot plate test, which measures the thermal sensation, pain sensation from the heat, was also, they have a much faster time to pain behavior when they were injected with IgG from long COVID patients. So these behavioral tests really reveal that these animals may be unable to coordinate their movement and balance themselves. So this would turn into some dizziness and inability to kind of maintain a posture, as well as this reduced muscle strength, as well as heightened thermal pain sensation. And to get down into more of the cellular details, Christopher Baker, who is a neuroscientist who joined our lab recently, did a detailed analysis of the nociceptor neuron ending in these animals. So using the same set of animals from which the IgG from healthy or the long COVID patients were possibly transferred to the mice. He used an animal that is fluorescently labeling these nociceptor neurons, so he can actually track the ending of these neurons using 2-photon microscopy. And using this, he's able to quantify the amount of these intraepidermal neuronal fibers using the 2-photon microscopy, and the collagen matrix shows up beneath the epidermal layer. And so this is what the picture looks like. So this is a fluorescently reporter mouse of SCN10A Cree, TD tomato, which labels the fibers fluorescently, so you can actually measure these things. This is up on the top row is a mice that were injected with healthy control IgG. And you don't see much difference in the fiber density within the first 8 days of injection. Whereas when you look at the bottom row, these are the animals that received IgG from long COVID patients. And they have much reduced fiber density in the epidermis. And if you measure these intraepidermal neuronal fiber volume, you see that the healthy control participants' IgG made very little difference, whereas the patients, long COVID patients' IgG reduced these volume as well as the number of fibers within the first day of injection. And many of these animals declined over time in these fiber numbers. So these experiments collectively suggest that the autoantibody or cross-reactive antibodies from patients is impacting the neuronal fibers. And this is typically seen for small fiber neuropathy models in the animals. So we think that that may be the reason why these animals have heightened pain sensitivity in their extremities. And finally, we also looked in the tissue damage and dysfunction. This is the hypothesis that the acute phase infection can trigger chronic changes in the tissue that may result in dysfunction. And this was a study that we published with Professor Michelle Monge from Stanford and her team. And this experimental setup really utilized our previous model in which we can confine the infection to a particular organ of interest. So by expressing human ACE2 only in the lung by intratracheal delivery of AAV vector expressing human ACE2, we are able to then infect the animal with SARS-CoV-2 2 weeks later. Infection is confined to the lung. And the infection is also transient. It doesn't make any sickness behavior. When within a week, we can't measure any virus anymore. Despite that, these animals develop significant changes, chronic changes in the brain, which is summarized by this really nice review that was written for this particular paper. For instance, the subcortical white matter had this elevated levels of this reactive microglia that then we think leads to further brain dysfunction by reducing the number of oligodendrocyte precursors and oligodendrocyte that leads to the loss of axon myelination. As well as we saw hippocampus neural inflammation that also resulted in this activation of microglia, resulting in reduced myelination as a result of reduction in oligodendrocyte number, as well as in the hippocampus, we see reduced neurogenesis. These are all expected to result in cognitive impairment in these animals. And this happened as early as 7 days, but then persisted for at least 7 weeks post-infection. And autopsies from humans who died with mild diseases for other causes, mild diseases of COVID also had reactive microglia that was elevated in white matter. And direct infection of CNS is not required to induce significant pathology because our model doesn't allow brain infection. There's no viral entry receptor in the brain. So this led us to hypothesize that the chronic changes in long-lived cells as microglia could be resulting in the prolonged symptoms of patients, especially the neurocognitive impairment. And so when you take all of these studies together, and like we're one of many other laboratories who are investigating these mechanisms, there are at least 3 papers demonstrating reactivation of EBV. So that could be one possible driver of long COVID, particularly in women, as I just mentioned. This, reactivating virus could be causing the pathology as a result of the virus infection or immune response against it. Secondly, we see evidence of not just autoantibodies, but functional autoantibodies that are cross-reactive in animals that are causing these significant deficits. So this may be also a driver in a subset of individual. We also see sort of a universal hormonal dysregulation in the level of cortisol, as well as sex hormones in males and females. That turned out to be the most correlating. The testosterone level is the strongest predictor and also the strongest driver of sex differences in the immune responses that we see. And finally, there is a lot more organ involvement and severity that's seen in females with long COVID. And the fact that they're really reporting distinct set of symptoms leads us to think that long COVID may be driven very differently between females and males, and we need to separately study them if we want to come up with the best treatment options for people with long COVID. So now that we have these features that are distinct in people with long COVID, we think that it is now time to be able to interrogate these pathways in vivo in humans in the form of randomized clinical trials and learn from these trials if you can measure the biomarkers before and after, or before, during, and after the treatment. For instance, there are many wonderful drugs that are available for autoimmune diseases, which can be repurposed to treating people with these autoantibodies. So immune targeting strategies, we can think of many different ones that can be tried. For the viral reservoir, we completed our Yale Paxlovid trial. We have not been unblinded, so we don't know what the results are yet, but shortly we'll find out how many people benefited from Paxlovid. And then we can also figure out which biomarkers correlate with the positive response to Paxlovid, so that in the future we might be able to selectively provide Paxlovid to those people with that set of biomarkers. We are also gearing up to starting a trial with Dr. Wes Eli from Vanderbilt, who is heading this very large randomized clinical trials called Reverse LC trial. This uses baricitinib, which is a JAK inhibitor, which will interfere with cytokine signaling. And we hope that will have a significant impact on reversing some of the tissue damage and chronic activation of immune cell types. And finally, we are also gearing up to start an antiviral trial for latent virus reactivation with Dr. David Petrino. This uses HIV drug, Truvada and Maraviroc, which inhibit the reverse transcriptase, but it also has some antiviral effects on Epstein-Barr virus, as well as reverse transcriptase from the endogenous retroviruses may be inhibited by this trial, the Truvada treatment. And Maraviroc is a CCR5 antagonist, which hopefully will interfere with infiltration of inflammatory monocytes and lymphocytes. So by combining these targeted strategy with biomarker identification, we should be able to learn a lot about what subset of patients have these drivers and what are the proper medicines that can be given to those people so that they have the best chances of recovery. I want to just end by acknowledging the amazing team of scientists that I get to work with every day, and the donations and funding from multiple different agencies that enabled us to do all these studies. And we're also now expanding our investigation to including ME/CFS patients, as well as chronic Lyme patients, to be able to identify head-to-head comparison of these different drivers of disease that may end up in a similar kind of post-acute phase of infection. And I want to end by thanking all the participants and patients without whom none of this insights can be gained. So I'm going to stop here and take questions if you have.
There's plenty of questions.
It was a fantastic talk.
Thank you so much. Thank you.
Everyone in here, thank you so much for connecting. We are with Dr. Akiko Iwasaki, and I am sure you know, but she's the actual president of the American Association of Immunologists. And I would like to congratulate Akiko as well, because she was awarded the 2024 Time 100 list, which means that she's one of the 100 most influential people of the year. So
thank you so much.
And we understand why, right? Like, she's done so much for patients with COVID. So thank you.
Thank you.
I actually have a question. I started asking a question, so I'm going to take the opportunity to ask you, Akiko.
Yes.
In thinking about the cause of those IgG antibodies, do you think there is originally a skewing because the infection would lead more probably to IgG being produced at the mucosal surface in the lungs, right? Do you think there is a problem there?
Oh, IgA?
Yes.
Yes. So yeah, that's a really good question. We actually have a parallel study looking at saliva because it's difficult to understand mucosal IgA without measuring mucosal surfaces. So we have that study ongoing, and we should be able to tell what's going on. As you suggest, Marianne, I think what's happening may be that these patients who develop long COVID, they're unable to clear the virus in early phases, which is then resulting in this, a post-acute phase of disease and likely an establishment of viral reservoir. So IgA would be a very important effector mechanism to combat that, but they may not have been able to generate that early on. So, but that's something that we are actively looking into.
Okay, thank you very much. Dr.
Yes.
Ivan, I think you had
I had a very specific question. So do you know if patients with long COVID tend to drink more? And is this associated then with the low cortisol levels?
Oh, drink water or?
Well, yeah, drink water
Yeah,
more.
I see. I'm not sure. We didn't survey how many
Well,
milliliters.
let me tell you that maybe you should.
Uh-huh.
That one may be one question that you should ask to to the patients.
Okay. Yeah, but so you're thinking that if their water consumption is up, the circulating level of cortisol and other factors should be lower,
Should be
right?
lower.
Yeah, but we didn't see changes in other factors though. Cortisol was sort of unique.
Okay.
So that's why we think it's more the pituitary problem. But yeah, that's something that we need to look into.
So then we have questions in some in Spanish, some in English. So I will maybe translate the next one, which Jennifer Hernandez was asking if it's possible that the reservoir for the virus is within the neurons.
Yeah, we can never say that it doesn't happen, right? But of all the different autopsies that are done and biopsies, it looks like neurons may not be a major reservoir. It looks like the intestine is as a reservoir, because many reports are now showing viral antigen and RNA in the intestinal mucosa, both in the epithelia and in the lamina propria. Also, there are, likely lymph nodes or some other, appendix. It's been shown to have viral RNA, as well as many, many organs. But seems like if you look at the PET scan that Tim Henrik did of activated T cells throughout the body of people with long COVID, they're finding elevated activated T cells in long COVID patients within the intestine and in the nose too. So maybe these are the places where the virus is too. We don't know exactly, but we're suspecting intestine is one of the major reservoirs.
So then there was somebody, Marta Resendiz, who was, oh no, hold on. Do you want to keep going, Mariam, while I'm,
Yes, sure.
okay.
So Victor Ruiz is asking, have hormone receptors or aromatase levels been evaluated in patients with long COVID, or its expression of IL-4 positive, IL-6 positive CD4 cells?
Yeah, excellent questions. We are looking into these levels. It's not trivial to look at the hormone receptor levels, but we are looking at aromatase expression levels. Again, like it's difficult to sample the right tissues in humans, right? We just have access to the blood and saliva right now. But we are also making animal models of all of these. So there we can do castration, we can do anything, to interfere with these levels. Yeah, so that's something that we're trying to do, is to draw causality between these hormone defects and the outcomes in these patients.
Thank you. Luis Donis is asking, have you observed whether pediatric patients are more susceptible to inflammatory responses in tropical diseases?
Oh, that's interesting. We don't know. We are also starting to look at pediatric long COVID patients. There, it's interesting because the older children, the adolescent age is where the increase in the rates of long COVID happens. And that, again, it relates to the sex hormones. So maybe this sort of puberty and sexual maturity is a driver, one of the drivers of these diseases. We have not studied tropical diseases, so it's hard for us to know if that's children are more susceptible or not. But I'd love to learn more if you have any ideas.
Thank you.
There is one question in the questions and answer.
Yes, I was going to go to that one.
Okay, so maybe I read it. It's from Lourdes Arriaga, and it's actually a two parts question. Do you think these post-virus infection syndromes may be a manifestation of premature aging of diverse systems, including the immune one? And then the second part is whether the increased production of cytokines could be rather a sign of cellular stress more than sustained inflammation.
Yeah, those are great questions, difficult to answer, right? For instance, it's been shown that COVID, the infection with SARS-CoV-2 does hasten the aging process. So if you look at the brain of people who had COVID, there's like a 7 years of healthy aging process that happens, that is hastened in people with who had COVID. So there seems to be sort of, CNS aging process that's speeding up after the COVID process. And also the UK Biobank study really shows the shrinkage of certain brain tissues post-COVID in people who've been followed, pre-COVID to post-COVID era. So that suggests that there may be increase in aging that's happening. Whether the premature immune aging is what's driving it, we don't know. And the other part of it is the cellular stress, whether that's driving the cytokine. Again, we don't know what's driving the cytokines in general. We think it could be either the persistent SARS-CoV-2 autoantigens, or it could be EBV reactivation, or it could be combination thereof. And of course, all of these events will be leading to cellular stress response. So it's hard to even dissociate what's going on. That's why the animal models are informative, because we can isolate and reduce these problems into each type and then look at the consequences.
Then there is a three-part comment and question here from Marta Resendiz, who says, well, hold on, I have to translate because it's in Spanish. And she says that she's in a research project where they are working the neuropsychological profile of young adults with long COVID. And then the next part of it is she has applied neuropsychological tests and she has found differences between the group with long COVID and the group without long COVID. And then she says that she finds a changes in the executive functions, whatever that is, important. So it's sort of a comment, I think, that is going along with what you mentioned too.
Yes, that's right. So we suspect that the animal phenotype that we see would lead to the reduction in the executive function of the brain. So, and then in humans, I didn't have a chance to explain other people's work where humans, when they do this functional MRI studies, they're seeing sort of elevated excess water content in the white matter of the brains of people with long COVID, again, consistent with cytokine secretion and increased infiltration of fluids into the brain. And so all of these things will then lead to neurocognitive deficits and, conditions that this person is referring to. So yeah, I think there's a connection there.
Thank you. I'll give the next one. Pablo Romero is saying hello and is asking, do you think that the autoantibodies found in long COVID patients could be similar to those of autoimmune vasculitis?
Great question. We are now looking at how these autoantibodies are actually causing these features that I explained to you. One possibility, for example, for the small fiber neuropathy is that the antibodies directly targeting the neuron fibers. Whether that's occurring or not, we don't know. How we're doing this is we're looking at cross-sections of different mouse tissues, staining with the IgG that's verified from patients, and trying to correlate the staining pattern to what's going on in vivo. Vasculitis is interesting because we and others have seen evidence for microclots as well as platelet activation and potential vascular engagement by the immune cells. So whether the autoantibodies are binding to vasculature or not, we don't know. Again, by doing this cross-section, we should be able to tell. So far, vasculature is not coming up was one of the key signals, but we'll see.
We have two questions that actually are asking the same thing. Is could PD-1 and TIM3 be considered as specific parameters to define long COVID? And the other question was, which markers are you using to identify CD4 exhausted T cells? So
yeah, basically those markers, PD-1 and TIM3, and TIGIT, we also stained together. But what we're, others, I should highlight Nadia Rohn's study from UCSF, where she showed that also exhausted T cells are elevated in patients with long COVID that are specific to SARS-CoV-2 antigens. So this means that not only are these sort of generally elevated, but also maybe reflecting chronic antigen stimulation by SARS-CoV-2 antigens. And whether these markers can serve as a potential biomarker for long COVID, that's questionable because not everyone has these exhausted T cells. As you saw, right, there's a huge variation in the percentage of these exhausted. Some of them have much higher levels and others have normal levels. So for it to be a biomarker, we want something that's going to stand out, that can be used to really dissociate people with long COVID. And cortisol level is one of those factors. But again, this needs to be validated in a much larger cohort. So yeah, I mean, collectively, yes, there are signatures, but we don't have like a single marker that says, okay, you have long COVID or you don't.
Thank you. I think we have time for a couple more questions. I see one that calls my attention. I may have missed it in your talk. Gustavo Pedraza is asking, have you done the IgG transfer experiment in both male and female mice?
Yeah, so we're doing those experiments. In the first set of experiment, we only used female mice, but we're also now going to compare male mice to see whether behavior is different. It's just that every experiment just sort of balloons into a huge cohort. But yeah, it's a very good question. Very good question.
Thank you. We have a lot of congratulations in the chat for you.
A lot of congratulations, exactly.
Thank you so much. I really appreciate it. Thank you.
Dr. Vianney Ortiz is asking, thanking you for your lecture, is asking if you expect that some patients may develop MS.
Oh, yeah. I suspect that they might. I mean, just as EBV did.
Yeah.
Maybe some people with long COVID might develop MS. As I mentioned, the general increase in autoimmune disease is seen in people after COVID. So many infections have been associated with autoimmune disease onset, and I'm afraid that SARS-CoV-2 is one of those infections. And we already see like this demyelination. It's not mediated by T cells or antibodies, but we see demyelination as a result of reduction in the oligodendrocyte loss, in the animal model. So if that's going to happen in humans, and there will be MS-like sort of feature clinically happening in these people. So hopefully we can detect and prevent these things from happening, but that's why like urgent research is required.
Thank you very much. I think it's going to be time for you to go to the airport.
Yes. Thank you.
We finished just on time then.
Just in time. I really appreciate everyone's questions and congratulations. Thank you so much.
No, thank you so much for sharing your so tight time with us. And I think that we at the Mexican Society of Immunology are really very honored of having you with us, at least as I say,
Thank
almost
you.
with us.
Thank you very much. Thank you. See you
And
soon.
Have a safe
Thank
night.
you.
Doctora
Thank you
Akiko.
so much for
Thank
everything.
you.
See
Thank
you
you.
Thank you
soon.
everyone to be with us also, and it was really a pleasure.
Thank you. Bye-bye.
Okay, bye-bye then.
Bye.
Bye.
I would like to remind those of you who are still here that we have events, I think, that are together with me at, ay, perdón, ya podemos regresar a
Mario, ya puedo regresar al español.
tenemos eventos con la Academia de Medicina esta semana, ¿cierto? El jueves y viernes.
El jueves y el viernes, sí. Entonces, bueno, pues igual, sí, por favor, o sea, los que están en la Ciudad de México podrían ir presencialmente, es de las 3 a las 6:30, algo así, de la tarde en el Centro Médico Nacional, en la
No,
sede de la
por
academia.
YouTube creo que se puede
O
ver.
por YouTube, exacto. Y los que están, pues, más lejos pueden ir virtualmente.
Muchas
Y
gracias.
seguir invitando para los nuevos seminarios, los siguientes seminarios que tenemos de la Sociedad Mexicana de Inmunología y que estén muy atentos a
Muy
ellos.
bien. Muy bien, muy bien. Pues muchas gracias a todos. Y pues nos vemos entonces pronto. Tenemos un seminario el próximo jueves, ¿verdad?
Con el doctor
Está
Polo, correcto,
el
para hablar
doctor
de
Polo.
globalización de la inmunología.
Exacto.
Sobre
Y
todo los
esto-
jóvenes
Exacto, sobre todo para los jóvenes es bien importante que vayan. Y en esta ocasión es el 23 de mayo, no va a ser el último jueves del mes, sino el 23 de mayo.
Muchas gracias.
Muy
Buen
bien,
día.
muchas gracias a todos y bueno, pues gracias por estar con nosotros. ¿Podemos parar el recording?